Hi, today I'll be talking about some aspects of molecular biology. First off, the central dogma. The central dogma is the flow of genetic information from DNA to RNA to protein, since genes identify the sequences of, or encodes, RNA, which in return encodes proteins. It is also, in other words, the process where the instructions of a DNA is converted into a functional product such as protein, which is known as gene expression. The process of DNA specifying the sequences of RNA is called transcription, which occurs in the nucleus of the cell. The transcripted mRNA will be transferred to the cytoplasm. There's three main stages that occur during transcription in both prokaryotes and eukaryotes: initiation, elongation, and termination. The first stage, initiation, is when the transcription bubble, the region of unwinding of the DNA double helix, is formed. It is also the binding of proteins and enzymes involved in the transcription at the promoter. In the second stage, elongation, the DNA strand that transcription proceeds is called the template strand, which the mRNA product is complementary to it, and almost identical to the non-template strand, which the thymine, the T, is replaced with the uracil, the U. During the process of elongation, RNA polymerase, an enzyme, conducts a similar process as to DNA replication, but the synthesized RNA strand isn't bound with its DNA template, unwinding and rewinding the DNA as the polymerase moves along the DNA. The last stage, termination, is when the RNA polymerase transcribes a sequence of DNA called the terminator. The transcription process ends here, and the mRNA detaches from the DNA. The process of translation converts nucleotide-based information into a protein product, as said before. Protein sequences contain 20 amino acids. A triple codon, a 3-nucleotide sequence combined corresponds to 1 amino acid, with a total of 64 combinations. Within the 64, there's 3 combinations which are stop codons, which are the termination of translation. There's also a combination called the start codon to initiate the translation. This genetic code is universal. The polypeptide chain or amino acid chain is formed as the mRNA moves to the ribosome. Lastly, I'll talk about the tools of synthetic biology: standardization, abstraction, and synthesis. Standardization has the aim to create sets of components which can be used in multiple systems and form different outcomes through recombinations. Abstraction is doing the job without getting into an unnecessary amount of details. Synthesis is the creation of the DNA complementary strand with both original strands acting as a template for it. And that's it for this video. Thank you for watching.